Okay, good evening, everyone. We are welcome to today's discussion class. And as you can see on the screen, our topic today is discussing a very interesting one, the dermatophytes. Okay, and we are going to be focusing on three major genera. We have the Trichophyton, we have the Microsporum, and then the Epidermophyton. Okay, so that's the major one we're going to be focusing today, and we're going to do justice to them. And as usual, it's going to be very simple, straight to forward, and then it's going to be very, very interesting, I hope. So, okay, so let's just get started. Okay. Okay, so what are dermatophytes? Like when you hear of dermatophytes, what actually comes to your mind? Dermatophytes are a group of closely related fungi that infect the keratinized tissues. And what are keratinized tissues? They are tissue that produces large amounts of the protein called keratin, which makes it strong and resistant to abrasion. So you could just say that they can be found on the skin, on the hair, and on the nails. So this keratinized tissue, let me just throw more light on it. They are proteins that usually produce on a particular part of the skin. And what they do is that they help to protect that part of the skin, okay? So okay, so what this keratin does is that it helps to produce that part of the body. So you can check the skin on your um on your nails, okay? You can see the skin on your scalp and then on your leg, okay? So you can see they have a particular and unique type of skin there, and it's because of this protein called keratin, which is strong and resistant to abrasion, okay? And also helps to protect that part of the skin from maybe from invading organisms or from anything that is going to cause it. Please, can you hear me now? Can everyone hear me? Sorry, I was a bit distracted.
Yes, it went
Yes.
off.
Okay.
But then we can hear you.
You are muted.
Oh, so sorry about that. So another specific characteristics of these dermatophytes is that they cause superficial fungi infections known as tinea or ringworm. I know at one point you must have heard of ringworm. It's like a general name for it. So they are named based on the affected body sites. Example is the tinea pedis. If you have heard of pedicure, you know that is something concerned with the feet or the leg area. Okay, so the ringworm, they are particularly named based on the area of the body they are affecting. Okay, so they also produce enzyme, the keratinase, which I've explained, that allow them to digest keratin and invade the outer layers of the skin, the hair shaft, or the nails. So what this dermatophyte actually does on the skin, because they actually like keratin, they use it for they actually utilize this protein on the body called keratin. So what they do is that they produce this enzyme called keratinase. So this keratinase digests the keratin on the skin, on the hair, and other parts of the body, and then they are able to invade and cause infections. Okay, so dermatophytes are occur worldwide based on geographical distribution, and for some species actually have a limited distribution. We're going to be seeing that when we're discussing specifically on each genera. Okay, so let's look on the ecology of the dermatophyte. That's the places where you can find them, or the natural sources and the mode of transmission. We have the one that are anthropophagic. These are majorly associated with human activities, okay? Maybe based on what like these are majorly concerned with human. Okay, they can be transmitted from man to man, maybe by close contact or through contaminated objects. Okay, then another one is called the zoophilic. They are usually associated with animals transmission. Okay, maybe from animal to man. Let's say, for example, from cat to a man. Maybe this dermatophyte they infect cats, and from cats they tend to infect the men. Okay, so then another one we have the zoophilic dermatophytes. They are found in the soil and are transmitted to man by direct exposure to the soil, maybe due to farming activities, gardening, and other activities. They tend to penetrate the tissues of man and then infect them. Okay, so just know that the knowledge of the species of dermatophytes and then the source of infection are important for proper treatment of patients and then control of the sources. Okay, to actually prevent further infections. Then another thing you have to know is, okay, I actually make a table for easy identification of some of the manifestations of the dermatophytes. Like I told you, they are generally called the tinea or ringworm. So we have different tinea infections. Okay, then the body side affected, like I said, they are being named according to the body side affected, then the common name that are being called, and then some of the causative agents like the species that actually causes these infections. For example, we have what we call the tinea capitis. Tinea capitis normally affect the scalp or and the hair. Okay, and the common name is the scalp ringworm. I'm going to be showing you a pictorial example of what I mean by this. Okay, so this I think this is just the most common, especially those times when we are growing up as a child in the village. You just find out maybe due to poor hygiene or just growing up in the village, and there's tendency of easy transmission of all these fungal infection. Okay, you just see something ringworm growing in your on your scalp. Okay, so uh, I've actually experienced that by growing up. Okay, these are the causative agents. We have the Trichophyton tolerance, we have the Microsporum canis. Okay, so we have the tinea corporis and noduli for the skin, the arm, the leg, the torso, and then the common name is the ringworm of the body. Then we have the tinea cruris, the growing area. We have the tinea pedis I've mentioned earlier, which actually affect the feet. Okay, we have the tinea unguum or onychomycosis, which actually affect the nails area. Okay, and they are called fungal nail infection. So you can just look through it and then try to master at least one or two, like at least three of them in case if you are asked in exam. Then some of the species, if you notice the most notable species or one that is very common is the Trichophyton. It can majorly be found in all of them, almost all of them, apart from the tinea capitis and then the tinea barbae. Okay, so it's a major and a very common dermatophyte species. Then moving on to the next one. This is a pictorial demonstration of the different tinea species I've mentioned before. We have the tinea capitis, which actually affect the scalp of the head. We have the tinea barbae of the beard area, especially among men. We have the tinea facial that actually occurs at the face, and then the tinea corporis, this one that affects the skin area. Okay, then let's move on to the three genera of the dermatophyte. Dermatophytes actually have three major genera, and that's where I'm going to be focusing on. We have the Trichophyton species, we have the Microsporum species, and then the Epidermophyton species. Then the key major difference between these three genera is that the Trichophyton, which is the most versatile one, like I explained before, is actually infects both the skin, the hair, and the nails, and it's versatile. Okay, then the Microsporum infects the skin and the hair area, but not the nails. They rarely affect the nails area. Then the Epidermophyton infects skin and nails, but not the hair. Just kindly know the difference, the key difference among the three genera, in case if you are asked to maybe right on the Trichophyton species. You just know that affects the three areas of the dermatophyte infestation, but the Microsporum is skin and hair, while the Epidermophyton is skin and nails. And the common ones among them is the skin, as you can see. All of them affect the skin. And unlike the Trichophyton, the Microsporum doesn't affect the nails, okay, or its bones. Then let's go back to the Trichophyton. The Trichophyton species are the most common dermatophytes and they are highly adapted to human hosts, often causing chronic infections. They infect the skin, hair, and nails, just like I've explained before. And they rarely cause subcutaneous infection in immunocompromised individuals, particularly patients with chronic malignous leukemia. Okay, so why we've mentioned that the dermatophytes actually affect the skin, the nails, and then the hair. So why is it that this trichophyton really cause subcutaneous infection in immunocompromised? Like in patients with the CML patient, the immune dysregulation allows the trichophyton species to normally confine to the stratum corneum to invade deeper tissues, especially when skin barrier integrity is lost. This leads to rare but severe forms like crayon or deep dermatosis. So what this means is that in patients with chronic malignant leukemia, what they do is that they are the ones like with the cast of blood, okay? So the areas on the skin, like the skin and the hair and the nails area, are already like disrupted due to this disease. And this Trichophyton species will then affect the deep tissue. Instead of affecting only the skin area, that is the subcutaneous part of the body, they invade the deeper tissues, okay, and start causing very severe and deep dermatitis. This is why they really um, affect the they really cause subcutaneous infection because most of the time the cutaneous area already affected, they already destabilized, and then the trichophyton actually go deeper inside the tissue to invade them and then cause deep dermatophytosis. Then the trichophyton rubrum is presently the most common cause of tinea in South Carolina. If you look at the chart I showed us before, there's a tinea, here this chart, sorry, the tinea infection table I showed, you notice that the Trichophyton rubrum is the most common causative agent among them, okay? Apart from the tinea capitis and the tinea barbae, like I've mentioned before. So it's a very common dermatophyte species that actually affects humans. So moving on to the common species of the Trichophyton, we have the Trichophyton rubrum, which I say is the most common cause of athlete foot, and then onychomycosis, which is a fungal infection of the nail. Then we have the Trichophyton metagrophrites, which causes most of the inflammatory lesions, and then the Trichophyton tossurans, which cause common cause of tinea capitis, especially in children. Okay, so these are just the microscopy and then the macroscopy. So what are the difference between microscopy and macroscopy? Can someone try? When you hear microscopy and then macroscopy, if you have read the mode 1 of material, I think you should actually have an idea of what microscopy and macroscopy is. Can someone try? Hello?
Hi.
Okay. Kemi, can you try?
Macroscopy is what we see physically.
Okay.
What we can see. The microscopy, what we can see with the naked eye.
Okay, so I think that just explained the literal meaning of it. Yeah, you are correct. So when you're talking of microscopy, especially in when you want to identify organism, so microscopy are just when you for example, when you use microscope to observe those features of that particular organism that cannot be seen with naked eyes, okay? Just like in a microscope. So microscopy. Then the macroscopy is actually when maybe you grow the organism in a culture media, then you have to observe with your naked eye. Okay, what are the features? Is it grow? Is it turning red? Is it turning blue? What's the color of the colony? That's macroscopy. You're actually observing with your naked eye on maybe on culture media. Okay, so when you do a macroscopy of this type of item, you notice that the cornea are large, which is called macroconidia. Just know that macro means large. Okay, then they are small, they are thin wall, and they have septates like 0 to 10 septates, which are like divisions. I'm going to show you what septate means. And they have a pencil shape. Okay, so these are just what we're going to observe on a microscope. And we have presence of numerous micromedia of different shapes. Then another one is that it takes 2 to 3 weeks to grow in culture. This is when you culture it on microbial media. It takes like 2 to 3 weeks to grow at a favorable temperature. And the colonies appear to be powdery to velvety. So when you observe the colonies that is being grown on the culture media, they tend to be powdery in nature. Then pigmentation, which is the color it shows on the colonies, so it varies from white to reddish on the surface. Let me just show you a bit of what I'm talking about so that it's not like I'm speaking English. So this is like a species of Trichophyton when it was grown on the media. Can you see that some are whitish to reddish depending on how long? Like you notice, we said that it grows like from 2 to 3 weeks. So depending on how long it has grown, it can go from whitish to reddish when grown on media. Then for the microscopy, you notice a large, smooth, thin-walled septate and pencil-shaped cornea. So this is what a cornea is. It's just like a reproductive part of this organism. Okay, so these are septates that have been separated. Like you can count, like these divisions or these, like it's looking like what is this, green beans, right? But it has like a division, so they are called separate. And if you look at the shape, they are pencil shaped. So this just a pictorial explanation of what I was explaining. Then let's go over to the Microsporum species, which is another genera of the dermatophytes. So they are most frequently associated with hair and skin infection, especially the tinea capitis in children. Okay, so they rarely affect the males, just like we've mentioned as a key feature of it. Then most of them are zoophilic, which is animal associated. They can be transferred from animals to man. Okay, so maybe one of the ringworm actually affects your cat. You have a cat as a pet. This can be transferred from it to you, which is called a zoophilic infection. Okay, and they could be easily identified on the scalp because infected hair fluoresces a bright green color when illuminated with UV emitting wounds light. So this is a very key feature of this Microsporum species, is that they actually show when you check them on a UV light as maybe a clinical investigation, they emit fluorescence, a bright green color. Okay, so maybe you are diagnosing a patient in the lab and you're trying to check which of the dermatophytes is even affecting this patient. Then you take them to a UV light to observe, and this is actually emit or shows a bright green color. You then be um, suspecting the Microsporum species, unlike other genera of the dermatophyte. Okay, so Microsporum canis is one of the most common dermatophyte species in humans. That is one of the species of this Microsporum species general. So the another species apart from the Microsporum canis is the Microsporum gypsum. So it is a soil dwelling and zoophilic. Okay, zoophilic means it can be found in the environment like in soil and other environmental habitation. Then for the microscopy and macroscopy, then the cornea are large, microcornea just like that's one we've explained. They are thick walled and they have spindle shaped, unlike the previous one we say they have a pencil shape, right? We say the Trichophyton has a pencil shape, but the Microsporum has a spindle-shaped conidia. Then the conidia are diagnostic hallmark under microscope, even for the Trichophyton species. You can use from the microconidia and identify them based on the shape. Maybe spindle shape, you know that this is a Microsporum species. When you see a pencil shape, you know that this is a Trichophyton species. Then the microconidia are scanty or absent, and when they are present, they are club shaped. So unlike in the Trichophyton where the microconidia are numerous, here the microconidia are scanty. Okay, the colonies are often cottonly or woolly. Okay, this is when you do a microscopy, when you go on the culture media, you notice that the colonies are cottonly or woolly. Then pigmentation varies from white to yellowish, while in Trichophyton it varies from white to reddish. You just know the key differences among them. I'm seeing some parts in the chat box and oh, sorry, my voice is low at some point. So sorry about that. Maybe I should just talk through the mic. But can you hear me now?
Yes.
Okay, so I can just talk through the mic for to be audible enough. Okay, so these are the macroscopy and microscopy for Microsporum species. Then this a pictorial view, the growth on culture media, which you see is vary from white to yellowish color. Then this is the microconidia, which you say is thick wall, spindle shape, and then it is multicellular. This is a kind of spindle shape, unlike the Trichophyton that is pencil shaped. Okay, hope you're noting down the key differences among them, just in case. Then the last but not the least species is the Epidermophyton species. So the Epidermophyton species, they infect the skin and nails and rarely the hair. Okay, and they contain only one pathogenic species among humans, which is the Epidermophyton floccosum. So it is anthropophilic, preferring human skin and nails. So they majorly transmit from man to man, okay, maybe through close contact or through other human activities. It can be anthropophilic, which means it's through human activities it can transfer from man to man. Okay, so for the microscopy and macroscopy, the macroconidia are numerous, smooth-walled, club-shaped, and multi-septate, usually in clusters. So if you notice the key differences in microconidia of all these genera is that they are numerous, smooth-walled, but in the shape are different. The Trichophyton has a pencil shape. The Microsporum has a spindle shape, while the Epidermophyton has a club shape. Do you get? So I'm just trying to note the key difference among them. Then the microconidia are absent. The microconidia are absent. You cannot find a microconidia when you do a microscopy for Epidermophyton. But in Microsporum, they are rare, like they are scanty. Okay, they are scanty. Why in Trichophyton they are numerous, they are plenty. So when you see plenty microconidia, just know that you're suspecting a Trichophyton species. But when you see a small microconidia growing, that means it is. Okay, so we are in microscopy and macroscopy of the Epidermophyton, and I was trying to let you know the difference between the three genera, especially when you come to the microscopy. Okay, so I've mentioned that the macronidia are numerous, smooth-walled, and club-shaped. Then they have multi-septate, usually in clusters. Then the microconidia are absent, unlike in the other two genera, and which helps to distinguish it from other genera. Very, very important. And they are readily identified by the thick bifurcated hyphae. So these hyphae, they are filamentous structure of fungi that branch into two, means two at the tip, creating a Y shape. Okay, then the colonies are yellow colored, cottony cultures with a leathery texture. So when you grow them on a media, this is what you're going to observe, a yellow colored colony. Then let me show you a pictorial view of what I've been explaining. This is when you grow the Epidermophyton flocosum on a culture media. This is what you are going to see. This is, you can see the colony is looking like a cotton-like structure. Then for the conidia, you observe under microscopy, they are bifurcated, which means they kind of separated into two at the tip. For example, this is a bifurcated hyphae with multiple smooth club-shaped micronidia. Okay, so these are the key differences. Just know the differences between the three genera. So generally, the dermatophyte treatment, you treat them with antifungal cream or antifungal drugs. Okay, for example, we have the tofnatate, which is a topical cream you can apply since it's a infection of the skin, maybe on the nails or on the scalp of the head. You can just apply on the affected area. We have ketoconazole. Okay, we have itraconazole, which is an oral antifungal drug. We have the terbinafine, we have the caspofungin, and then the crisofovin, which is majorly for scalp and nail. So these are just the treatments that can be recommended for someone that is battling with fungal infections. Then we have something called the dermatophytic reaction. Okay, I saw it in the material and I feel it's very important for us to discuss on it in case they ask you what is called a dermatophytic reaction. All these microbiology terms make it so bite your tongue. So it's a hypersensitivity immune response that occurs from the primary site of a dermatophyte infection. So what is this is actually explaining is that sorry, I'm not seeing my screen. Okay. So what this is actually trying to explain is that sometimes when you have a fungal infection, particularly dermatophyte, maybe is occurring at the leg, okay, you start feeling a secondary lesion at your in your hand. I don't know if this has actually happened to someone. You start feeling another maybe irritation, itching, inflammation, or burning sensation in your hand. This is due to a hypersensitive immune response of what is happening at the primary infection site, maybe at the leg. Do you get? It's called a dermatophytic infection reaction. Okay, so one of the a unique feature of this is that when you actually check for fungal infection at that secondary lesion, which is on the hand, you not see any fungal reaction. But when the primary infection sites have been treated, the secondary lesion will be resolved. Okay, like I said on the slide there, this secondary lesion will not respond to any topical treatment or any antifungal, but they will resolve if the primary infection is successfully treated. I hope you understand what I mean by this dermatophytic reaction. Do you understand what I just explained? Please, if you don't.
No, there was a break. Can you please explain again?
Okay, I'm saying that it is a hypersensitivity immune response that occurs away from the primary site of infection. So what this means is that, for example, you have tinea pedis that is occurring at the leg, at the feet. Then you start noticing another reaction in your hand. Okay, maybe it can be itching, your hand will be itching you, or it can be maybe inflammation or burning sensation.
We can't hear you.
Oh my god, can you hear me now? Network is really messing up.
Yes, so
The thing is, it's actually breaking. So when you say one,
yes,
it goes off
but I
before you say
understood
the
what you.
next thing.
Oh, that is network. It just finished raining here on my side, so I don't know if it's affecting the network.
Yes, you mean the secondary lesion. Even if you did not treat it, it will go,
It will go provided you
right?
treat the
Yes,
primary infection. Yes.
in the primary, yes.
Yes, so
That's
that's
what
what
I
I mean by
understood.
yes. So please, have everyone understood it? So I'll go over to the next one.
Yeah.
Okay, so let us go over. I just pray the network will have mention of this night. Then, okay, so for the three genera that we've discussed today, these are just the summary of everything I've explained. We have the dermatophytes. They are a group of fungi that causes skin infections in both humans and animals. The major three genera that we explained and in context of our material is the Trichophyton species, we have the Microsporum species, we have the Epidermophyton species. Okay, and then these are the key features and then the key differences between them. Based on the macronidia, the Trichophyton have a pencil shaped, then the Microsporum has a spindle shape, while the Epidermophyton has a club shape. Then micronidia feature in Trichophyton they are numerous. In Microsporum, they are few or scanty, while in Epidermophyton, they are absent. They're not there at all. When it comes to hair inversion, Trichophyton affect the hair, Microsporum affect the hair, while Epidermophyton doesn't affect the hair. But in nail involvement, Trichophyton affect the nails, the Microsporum rarely or doesn't affect the nails, while the Epidermophyton affect the nails. And you know for the skin, all of them actually affect the skin. Okay, for wound gland reaction is very rare in Trichophyton, which is a very like good trying for diagnostic investigation for this genera. You can this wound lamp reaction just placing them on a UV light, and when maybe any of the organism fluorescence or show a bright color, just like the microsporum, which I mentioned that they show a bright green color, so they can be used in identifying them. It's very rare for trichophyton to show a positive reaction for wound lamp. The microsporum shows a positive reaction, especially the microsporum canis, while in Epidermophyton they are negative. Okay, so these are the summary of everything I've tried to explain above. And if you have any question at this point for the dermatophyte, just let me know. So maybe I can go over it again, or we'll just tackle the question. Or if you don't have, we'll continue to the next topic. Is there any question at this point? Let me know.
Yes.
Okay.
Yes, I want to ask you.
Okay.
But it's not only on dermatophyte. I just want to ask generally.
Okay.
It seems if they're asking questions, they ask sometimes on epidemiology. So I want to ask, is it the same with the geographical distribution?
Yes.
Like you can say it is distributed. I notice it's like it's the same thing.
Yes. When they're talking of epidemiology, they're just trying to ask you, okay, how does this disease occur, or where does it occur? How does it occur? Who does it actually affect? Okay. So is based on if they ask you the epidemiology, you can say the mode of transmission, what the disease is actually is, what is this disease? Like if you notice most of the slide I present, I actually mention okay, the dermatophyte, they are fungal infection that affects the human, the skin, the nails, and then the scalp. Okay, then you can also mention for the epidemiology, they are being transmitted through anthropogenic, zoophilic, or through what's the last thing we mentioned? So these are all epidemiology. Where do they affect? Do they affect the tropical or the subtropical region? How do they affect man? What is the mode of transmission? Okay, is it through animal to man, or through man to man, or environment to man? So these are just epidemiology, trying to ask you what's the meaning of this disease? How do they occur, or how do they transmit to another? I hope this answers what you're asking.
Yes.
Okay.
Your voice is breaking, but I understood. Thank you.
God, what's this network? God help us, please.
This network, even here.
Okay, please, is there any other question?